So I just got back from my genetics lab and there's an alleged hurricane outside, but I don't care because I was in my genetics lab and there were a whole bunch of people that had an issue with the 3-point test cross, and I just felt really frustrated because I got it, and I felt like yes, it can be a bit complicated, but if it clicks for you, it will click for you. And so I'm just gonna make this video so I can never get lost on it ever again, and if anybody needs it, it'll be helpful. So, a 3-point test cross, first I'm going to explain what it's going to be for. So, a 3-point test cross is basically trying to find out what traits are on what point in a chromosome. So, basically, all it is location detection. So, this is a chromosome, right? And just think of it as a road. Just think of it as you're driving from one point to another. So, if you're trying to find the location of your house or the location of a tree, or you're trying to find the location of your significant other, right? You're gonna have specific markers on this road that will help you an indication of where each thing is. So, for example, if you have multiple roads, there might be, an apple here. I don't know what that is. There might be an orange, and then there might be a star, in which case you would have to perform a linkage test with, with this, compare it to the apple, and if there's a significant, if there is a significance in the ratio, then that means that it will be on this chromosome. And for example, if you cross this, like your significant other's location with the apple, and you get a 1-to-1-to-1-to-1 ratio, it's not going to be linked. The genetics not going to be linked. But if you cross your lover's location with the star, and it gives you like a really huge skewed result, then it's going to be on there. But today we are not going to be talking about how to perform that test, but we are going to be talking about how to perform a 3-point test, or a 3-point test cross. And this involves 3 traits or 3 alleles that will mark that trait. So usually in genetics, you're going to talk about phenotypes, genotypes. We're not going to deal with genotypes in this type of test cross, but we are going to deal with alleles, which you're going to have to assign a random allele. It can be uppercase, lowercase, it don't really matter. All matters is you are looking at this. So how a 3-point test cross, basically the point of a 3-point test cross is to determine where a trait or an allele is on a chromosome and how far it is as compared to another trait on that same chromosome. So for example, right here, this is the chromosome. So you have 3 points. So this point could literally be the allele that, or this could be the point where this could be the point that codes for body type. This could be the point that codes for color of your body, and then this could be like hair color, for example. Then you don't know exactly which one is which, but in determining in 3-point test cross, you can find 3 traits in where they are located on the chromosome in relation to each other. So that's basically what we're going to be doing. I have already written out a result test for this. So what the result is usually you're given it for an example, for like a question. You're going to be given probably 8 different types of data points, because why? We're talking about 8 F2 offspring. So if you have a parent's generation with 3 offspring, or with 3 like traits, I guess, you're going to have an F1 generation with 2 types of F1, but your F2 generation is going to have 8 because of how many traits you have, So there's different recombination types. You're going to determine which So the location of each trait on a chromosome will help you determine whether or not it is easy to be it's going to help you determine if it's going to be more likely or less likely to recombine with another. So for example, if these are two chromosomes and hair color, body type, and then hair color, body type. So like these two, since hair color and body color are going to be super close on each other, it's most likely not going to mix up and recombine with the other chromosome just because they're so close to each other and they would just prefer to be closer to each other. However, if this is so far apart, it's going to be more likely to recombine with something else that's right here. So then when these two things cross, like you see how it'll cross. Most likely this will transfer to this and this will transfer to this. But this is less likely since they're so close to each other on the same chromosome. Okay, so now that I'm finished talking about that, we're looking at F2 offspring from these three traits. We don't know who the parents are, we don't know who the F1 is. We are looking at the F2 offspring from the generation. So most likely what what happened here is that there's one parent that is homozygous everything for wild type, and then you have another parent that is like trait A, trait B, and trait C, right? And then most likely your F1 will be all the same with regards to whatever's dominant and whatever's recessive. So for example, if your first parent is wild type homozygous and your other parent is homozygous for the mutant traits, assuming that the wild type is dominant, then your F1 will all be showing the wild type traits. However, they're going to be heterozygous in genotype for both traits, if you know what I mean. But you don't have to really know that. The results that you see on this board right here are most likely going to be given to you during a question. So this is going to be 8 different types of recombinations. So most likely what happened here is you had a parent generation and then you had an F1 generation. So most likely you have crossed a parent with wild type for all 3 traits, and then you have crossed a parent that is all 3 traits mutant, right? And both parents are homozygous for whatever traits that they are sharing. F1 will be heterozygous for the traits, so it'll look like this. However, whichever trait is dominant, either the wild type or the mutant trait, this F1 will be expressing. So for example, if the wild type is the dominant one, which is most likely the case, usually the wild type is dominant. So if the wild type is dominant, then all the F1s are going to be heterozygous for the traits. However, they're only going to show the wild type traits, assuming that this is simple Mendelian dominance. And if it's incomplete, then whatever. We're not really going to be talking about that right now. We're just talking about the 3-point test cross. So just know that you're looking at the F2 generation for right here. So to find out which allele is on the chromosome that I have drawn right here, you're going to first look at your data point and you're going to rewrite the data points as the allele. So you don't really need the genotype for any of this, but you're going to assign the allele. So right here we have wild, wild, wild. I'm going to write plus, 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 because that's usually the allele for wild type. So we have 14 for that one. Wild, green, wild. So I'm going to do plus, we'll put G for green, and then another plus for wild, and there's 120 of those. And then wild, green, stubble, we're going to put S for the stubble, then 117. So right now what we have so far is plus, 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 plus G plus, and plus GS. Curled wild, wild, we have C, and then plus, plus, and then there's 115 for that. Again, these data points are given to you, so this is randomly generated or just given to you as a problem. This is one of my example problems, so that's why I have that. But anyways, now we have curled green wild, CG plus, that's 1,227. And then you have curled green stubble, there's 8 of those, so I did CGS for that one. Curled wild stubble, C plus S, then there's 103 of those. And then wild stubble, so plus S, and there's 1,258 of those. So these are just the traits of a fly. So there's either a curled wing, a green body, and then stubble body, I guess. And then the alternate ones for those are wild wing, wild body, wild wing shape, or body shape. So anyways, I already wrote all the alleles for this one. So there should be 3 alleles for 3 traits, and then the corresponding numbers next to it. So you're gonna see that these numbers are very skewed. It's obviously not a 1-to-1 ratio, but it's very, very skewed. One big trick that you could do, what you're gonna do is you're gonna group them into groups, like groups of 2 for pairs. And a nice little trick to do that is you could look at the numbers. So if they're closer to each other, they're going to correspond to each other. For right here, CG plus is going to correspond with plus S, because they're both like in the thousands right here, as you can see. Not all the time you'll see like skewed results like this. Sometimes it'll be like 30, 36, 35, 32. You're not going to be able to find out. So how you can test to see if they actually go together or not is by writing them together. So right here I have CG plus, and then I have plus S. So that's 1,227 and then 1,258. I suspect that they go together, but how you really know though is if you look right here, C plus G plus S. None of them are the same. So you see, this is C and plus, G and plus an S. So there's one of each type of allele, and there's no like duplication. So if, for example, I'm gonna make this wrong, but I'm gonna test plus S and plus. So I'm gonna suspect that these are the ones that I should pair together. These are actually not paired together because you can't have 2 same allele for the recombination pair. So there's a whole bunch of scientific information that goes into this. I'm just gonna show you how to work it. You can follow this if you want to work it, but it helps a lot more if you understand the mechanisms behind it. So first, so we just paired these ones that have the highest number. This one's gonna be your parent phenotype. So we're gonna label that with a P. This is very important for the next step. But now we're gonna just pair the rest just to make it easier to look at. So the other 2 that are kind of low will be plus and CGS. Look, the phenotypes line up. There's one of each. You don't need to worry about duplication. And then you have 14, 8. So you already have this one, and this one. Now you have to pair these 4. They're kind of close to each other. You got 120, 117, 115, 103. What you're gonna do again, like I said, you're gonna look at the genotypes, which ones kind of don't match up with another. So CT or C plus, G plus, S plus. So now you know that these 2 go together. T plus GS, C plus plus, and then that's 117 and 115. Cross those out, and that leaves us with plus G plus and C plus S. All right, so we have successfully rewrote all of these allele recombinations. Now we're gonna just assign. So we already assigned the parent pair. This one's going to be your double recombination. So I'm gonna put DC for that one because it's a lot less likely for them to double switch up because again, they're linked alleles. However, they can switch up once. So there's 4 types of those. So we paired these 2 in the middle. They're both going to be the single recombination ones. So this top one with the most amount of offspring, that's the parent phenotype, so that's the P group. There's 2 SC groups because they're the middle ones, and then there's 1 DC group. All right, so we already assigned the alleles, and then we found the parent phenotype. So now we're gonna look at the parents and the double combination. So we're gonna find the middle allele right here. What does middle allele even mean? So again, like I said, we are actually trying to assign locations or like a locus for each allele. So we have one that's right here, one that's right here, and one that's like right here. So where's the curled locus? Where's the green locus? And where's the stub locus? It could be curled, could be right here, you know, curled could be right here, or it could be right here, or it could be right here. All you know is there's 1 gene per locus, and it could literally be like this. So in this case that I wrote it like this, G is in the middle, but it's not always going to be in the middle. So it could be C right here, G right here, S, G, S, C, C, S, G, like you don't know. So what you need to know before anything is which allele is in the middle, which one is not on the outside but in the middle. All you need to know is which one is in the middle, and then you can assign one is right here. Like, so for example, if G is in the middle, you know C is right here, and then S is right here, or you know S is right here or C is right here. It doesn't really matter, just the chromosome. You could flip it around if it's, you know, not to your liking. But all you need to know is the most important one is what allele is in the middle. So how you know the allele that's in the middle is by looking at the parent and the double recombination. So I'm gonna kind of like rewrite the parent and double recombination so you can see. So CG plus S. This is the parent. And then the double combination, you got plus CGS. And then this is the double recombination. Okay. So looking at this, which allele changes from, this is kind of complicated before you, like, if you haven't figured this out, then this might be like one of the most complicated parts to figure out. But once you figure it out, it should go pretty smoothly. So you see right here, CG plus. Down here, you got to look at these two as groups, right? So these groups have 2 people, like 2 types of people. So you got plus S and then plus. So let's just think of that as a girl with blonde hair, blue eyes, and a purse. And this one has blonde hair, blue eyes, and a pair of glittery shoes. Whoa, what's the thing that changed in between these 2 groups? Well, the last thing. This girl had, what did I say? It don't matter. But the last trait that I said, this girl had glittery shoes and this girl had a purse. So that's the last allele is the one that's changing. So it is switched up, but it doesn't really matter which one is on top, which one on the bottom. If you want to, since this one, the plus S is on the bottom, you could literally just CGS plus. There you go. One's on the top, one's on the bottom. The ones that are similar. So up top you see CG plus. Up top down here you see CGS. The red flag that you're seeing is that last allele down here, plus S, plus. Again, that red flag that you're seeing is on the bottom. So what trait is responsible for that allele? Well, stubble is, because S. So now you know that stubble is going to be in the middle. Woo! That's like already 50% of the question figured out. So now that you know that stubble's in the middle, you can either put curled or green on either of these ends. It doesn't really matter. But I'm going to put curled here, and then I'm going to put green. If you put it the other way around, that's fine too. It doesn't matter. But all that matters is that you have put stubble in the middle, right? Now you know that it's your middle allele. All right. Now what we're going to do, just to make things look a bit more like easy to determine, you're going to put the stubble allele in the middle. So you see how like right here I have written it on the end? We're actually going to we're going to switch the middle to the outside, or just however you wrote it, you know? So if I put green up here, we're going to have to do a lot more rearrangement, but since I put CSG right here, I wrote CGS, so I only have to switch the S and the G to make it match whatever I have put up here. So we were testing C, S, and G. I'm gonna rewrite everything. So for the parent, it was CG plus. I'm gonna put C plus G. And then for plus plus S, I'm gonna put plus S plus, because the S always has to be in the middle. And then for plus GS, I'm gonna put plus SG. For C plus plus, I'm gonna put C plus plus, nothing really changes. And then for plus G plus, it's gonna be plus plus G. For C plus S, it's gonna be CS plus. For plus plus plus, it's gonna be plus plus plus. And then for CGS, it's gonna be CSG. And then just copy the numbers. Again, it's just the same exact thing. It's just the order of the alleles is different. And none of these like allele orders is permanent until you have figured out the stub, like whatever alleles in the middle. And we found out that stubble's in the middle, so now you know that this is how it is. And it could also be, again, right here, G plus C. But G plus C is the same thing as C plus G, you know? So it don't really matter. Okay, so we have already rewritten the alleles after we've determined the middle allele. Now we're gonna find the needed recombination values. So right now, we're going to be determining the distance from this to this, and then the distance from this to this. And you can also find the distance from here all the way here. So now that we know the location of the traits, you just need to find out what the like different distances are between each, you know? So if this is your house, your mom's house, your friend's house, right now we're gonna find the distance between your house and your mom's house, and then your mom's house to your friend's house, and then also to your house, from your house to your friend's house. Basically, like this is literally just a map, but just a bit more complicated because we're dealing with genetics here. So we have these like data points. First, we're gonna find the distance from C to S, right? Like I said, CSG, so we're gonna find the first distance from C to S, and then now we're gonna find the distance from S to G, and then we're gonna find the distance from C all the way to G, right? These are basically the last 3 steps you need to do in a 3-point test cross, literally. So these are the 3 last steps you need to do. Okay, so I can't explain to you why, but I know how it works. So if you're going to find the distance from C to S, you're going to look at the parents. So the parents, I guess like allele combination was C plus, because the plus is where the S is supposed to be, right? So the parent is C plus, so you have to find a different combination of that. So for example, since it's C plus, you're going to be looking for CS. And then from down here, since it's TS, you're going to be looking for TT, or not T. When I say T, I mean plus, sorry. So you're going to be looking for CS and TT, or plus plus. Since the parent was already C plus and plus S, you know, you're not looking for that. And I'll explain maybe a little bit more of that later. So, and then from S to G, since the parents was from S to G, since the parents was plus and G, you're going to be looking for S, and sorry, give me a second. Give me. Oh no. You're gonna be looking for SG. My bad, and then plus plus. That's what I think. Yes, I'm right. Okay. So the reason why I say this is, okay, so now you're gonna look for CS and plus plus. This one doesn't have CS and plus plus. This one doesn't have CS. CS no. Oh, look, there's CS and plus plus. So you're gonna put 103 plus 120. All right. Does this have CS and plus plus? Yes, it does. And then you're gonna add 14 plus 8. And you divide that by the total, which I believe the total number of offspring here was 2,962, right? Okay, and then you're gonna divide that by 2,962. You're adding these offspring because they have the recombination types. So since these parents was C plus, if it recombined, it would be CS, right? Because yeah, you wouldn't need to count the C plus for anything. So when you're adding these numbers in this second step of the first problem, I guess, why would you need to add the parents? You wouldn't want to add the parents. You're trying to figure out the recombination. You're trying to figure out the recombination rate, or like the probability of recombining. So you wouldn't need to add the parent generation to that. So that's literally what you're gonna do. You're gonna add the 2 or the 4 kids that you saw that had the recombination traits, and then you're gonna divide that by the total number of offspring. So here, again, like I said, taking into consideration what I just said before, if you had S or if you are trying to find the distance from S to G and your parents was plus G for that trait, then you would want to look for SG and plus plus. Now, if you're looking at like something that says CSG, if the parents were CSG and you're trying to find the difference between S and G, you would be looking for S plus and plus G, you know, in comparison to this problem. But this problem is not like that. This problem, the parents were plus and G, so you're looking for SG plus plus. So where can you find SG and plus plus? Well, right here you can find SG and plus plus. So what you can do is add 117 plus 115, and then down here you see S plus G, no, plus plus and SG right there. Plus 14 plus 8. Again, whatever's on top and whatever's on the bottom does not matter. As long as there's a trait in each group, you add that group to the equation. So again, that you divide by 2,962. I am going to just do the math really quick. Give me just a second. 103 plus 120 plus 14 plus 8. That's 245. 245 divided by 2,962. So that equals 0.0827. Okay. 117 plus 115 plus 14 plus 8. That equals 254 divided by 2,962. And that equals 0.0857. So what you're going to do is you're going to convert this to percentage. So this is going to be 8.27. And then this is going to be 8.57. This percentages, you can just basically convert the percentage to MAP units. So 8.27% is also 8.27 MAP units. You can write that as MU, or you can write 8.27 CM. CM being capital because they're centimorgans. So yeah, it's not centimeters. If you write CM like lowercase, it's going to be centimeters. So 8.57 down here for S and G, that equals 8.57 MU, or 8.57 centimorgans. Okay, so how do you figure out the distance between C and G? Because we already figured out C and S and then S and G. Well, this is like elementary school math. So if C is right here, S is right here, G is right here, we found out this distance, and we found out this distance, then all you have to do is just add them together. So that would be 8.27 plus 8.57. Let me add that. That equals 16.84. And don't ask me if I'm wrong or right, because I already checked with my, this is my assignment, so I just am reworking it for you to see. So it will be, from here to here is going to be 8.27, and then from here to here is going to be 8.57, and that total distance is going to be 16.84. So that's literally it. Those are the answers for the 3-point test class. It's not that complicated, but if you like, I feel like the thing that I would recommend is just to keep practicing the thing that will mess you up the most is finding out which allele is in the middle, because you're like, okay, well, the G allele is in the middle, is it not? Okay, that's not what you're looking at. You're looking at the combination between all of these, between the parent and the double recombination generations, and you're finding out which allele is changing in between all 4 of them. And that, in this case, was the stubble allele, which is that last one, which means the stubble allele was in the middle. That is the first thing that you might get confused on. The second thing that you're getting confused on is the recombination thing to look for right here in this like last step. If the parent had a certain, I guess, a certain pattern, so for right here, it was C+, you're not going to look for C+, you're going to look for CS, because that's the recombination combination. That sounds really redundant, but that's what I mean. You look for CS and plus plus. And again, since the parent was plus and G, you're going to look for SG and plus plus, because that's the recombination. You're not going to be looking for the parent to like determine whether or not like it's like whether or not recombination will occur, because well, clearly recombination didn't occur for the most popular or like the most populated type of offspring. So of course you're not going to be ever adding these ever like these values to your equation, because it's the parent like phenotype. If the parent phenotype is going to be expressed, then obviously it's not going to recombine. What you're going to be, the 3, I guess like there's 3 points to the 3-point test cross. There's 3 purposes, and you need like knowing these purposes will definitely help you figure out like, you know, what you're looking for and like doing it correctly. So the first purpose is to find out which locus each allele is on and how far apart they're on. The second point to a 3-point test cross is finding out the percentage, like the probability of a recombination for any of these 3 traits. And I guess the third point is just to look at like, okay, which traits, which phenotypes are the parents, and which phenotypes are like, as it gets less and less likely, then that's the more recombination it is, because since it is a gene that's linked to another, because again, like I said, the genes that are closer to each other are less likely to, you know, recombine, and they're more likely to just stay and persist among generations, and the ones that are farther away are going to be more likely switched up. So right here, again, you see the parent generation is very, very high. They're in the thousands when everybody else is pretty much in the hundreds. So that those genes are not going to recombine as much, or yeah, they're not going to recombine as much as the one down here. And you see like it keeps getting less and less, because single recombination is very unlikely, and then double recombination is even less likely. So yeah, I hope that helped.